So welcome to you all, and thank you for joining me today to talk about the Pathfinder Target Essentiality Assay. This assay that we developed here at Horizon will enable you to identify genes essential for the growth of specific cancer cell lines. It can be used to definitively resolve ambiguous screening results, and it can also provide information on target suitability by testing essentiality in either normal cells or in cancer subtypes different to the proposed patient population. But first, let's talk about traditional methods of target validation. A typical approach to both target ID and target validation is the use of RNAi, knocking down the target and looking for phenotypic effects. However, it's not without its weaknesses. While it's easy to use, it's often challenging to achieve a complete knockdown of expression, and a residual 5 to 10% of a transcript can result in masked phenotypes. Further to this, as the study highlighted here demonstrates, RNAi screens are often difficult to reproduce. Here we can see 3 screens looking for HIV host factors with an overlap of just 3 genes between all 3 studies. Hence, there's a very real risk of false positives or negatives that are inherent to this technology. One of the most widely accepted caveats to using RNAi is that there can be significant off-target effects. These off-targets can be difficult to predict as they don't often correlate with any blast scores. There are known sequence-independent off-target effects. Exogenous expression of shRNA can overload the Drosha dicer machinery and can interfere with normal microRNA processing. Also, introduction of a large amount of RNAi into a cell can displace exogenous microRNAs from the RISC, which alters normal patterns of gene expression. Of course, there are also well-known sequence-dependent off-target effects. The seed sequence, which is a known driver of sequence-specific off-target effects, we can get complementarity between the 5' region of the shRNA and the 3' UTR of the mRNA. And this means that a single shRNA can actually affect hundreds of messenger RNAs. Moreover, with shRNA, some imprecise hairpin processing can lead to the production of multiple siRNAs, each with its own seed sequence, increasing further the amount of off-target effects. So, can we find a better way to ID our targets? The discovery of CRISPR-Cas9 has revolutionised target ID. The CRISPR-Cas9 machinery can be used to generate complete genetic knockouts, overcoming some of the false negatives due to incomplete knockdown that we see in shRNA screening. The technology relies on a single guide RNA sequence to direct the enzyme Cas9 to the gene of interest where it can introduce a double-strand break. Break is then repaired by non-homologous end joining, which can result in insertions or deletions at the cut site. The occurrence of any frame shift mutations results in knockout of the gene of interest. So how are we using CRISPR-Cas9 for target validation at Horizon? CRISPR-Cas9 is thought to have less severe off-target effects than shRNA, and complete knockout of the target can overcome some of the threshold effects that we see in shRNA experiments. However, it's not without its own issues. To help explain this, I'm showing some model data comparing shRNA and sgRNA. The parental line, which we can see here in red, grows exponentially with a doubling time of 24 hours. Cells infected with shRNA, which are these here in green, have a reduced proliferation rate, which increases the doubling time to about 72 hours. However, cells infected with sgRNA in blue have lost the essential target in probably 80% of the cells. However, there's a residual 20% of the remaining population which continues to grow with the same 24-hour division time as the parental line, and eventually will reach confluence. CRISPR-Cas9 relies on gene editing by non-homologous end joining, which means that only 2 out of 3 repairs will cause a frame shift mutation. Therefore, any in-frame mutations may not fully disrupt the gene. However, it's not possible to determine whether this has occurred in a heterogeneous population. In order to overcome this problem, we've used our vast experience in cell line engineering and the workflow required to analyse edited cells at a clonal level to develop our Pathfinder Target Essentiality Assay. In this assay, we target Cas9 to the gene of interest and then culture colonies in a single cell dilution. We can analyse hundreds of colonies to determine whether any clones can be recovered that possess a complete genetic knockout. If either no or very few colonies contain frame-shift knockouts on all alleles, the target is deemed essential in that particular cell line. So to explain the requirements of the assay in more detail, this assay requires on being able to screen hundreds of colonies, and therefore the cell lines of choice must robustly grow from single cells. We have a list of 53 cell lines that we've pre-evaluated for use in this assay. However, we can evaluate any cell line for suitability. Secondly, the guide RNA must efficiently direct Cas9 nuclease activity to the DNA. And if you do not already know of an efficient guide RNA, then we can design and evaluate them for you. We've designed this standard assay to be suitable for cell lines where the copy number of your gene of interest is about 2 or 3. However, if the copy number of your gene of interest is higher, then there are ways that we can overcome this. We would recommend increasing the number of colonies that we screen to account for the expected increased diversity of allele editing combinations. So I can now outline the assay process using an essential gene as an example. So the first step in the process is to determine a restriction enzyme that we can use in the assay. The ideal enzyme spans the Cas9 cut site, which, as we know, is about 3 nucleotides up from the PAM. And in this example, we've used HPY188. Your cell lines of choice will be infected with the sgRNA in an all-in-one vector. We are using the p-lenti CRISPR vector from the Zhang lab, which is referenced in this paper by Sanjana et al. Now, the infected population is then cultured in the presence of puromycin for 2 to 3 weeks to ensure all alleles have been fully edited, and then the cells are plated as single cells and allowed to grow. And then once the colonies have reached a sufficient size, they're harvested and we perform a PCR and determine editing by a restriction digest assay. Using this method, we can evaluate whether heterozygous or homozygous editing has occurred. So here's some model data to explain this part of the assay. The digested PCR products are resolved by agarose gel electrophoresis, and the migration pattern of the bands indicates whether editing has occurred. For example, in these lanes labelled with a yellow star, as you can see here, all of the PCR product has been digested, meaning that the restriction digest site is intact in all alleles, and therefore it's unlikely editing has occurred in these colonies. In these lanes labelled with the orange stars, here in the middle, we have a proportion of digested and undigested product. This indicates that the colonies are heterogeneous for editing. And in this lane labelled with the blue star, none of the PCR product has been digested by the restriction enzyme, indicating that editing has occurred on all of the alleles. So to analyse all of this data, we allocate the colonies to each category, and that enables us to visualise the overall data, as you can see in this graph here on the right. So now here's an example of some real data that we generated using a target in 2 cell lines, one that was known to be dependent on the target and one that we knew that was independent. So in this assay, we analysed approximately 350 colonies for the independent line. However, only analysed just over 50 for the dependent line. This line was really dependent on this target, and we actually recovered fewer colonies overall. So in the independent line, which is on the left of the screen, the gel image shows quite nicely that all the colonies have had some editing. And when we collate all the data in the graph, we can see that there are actually no unedited colonies here in this last bar on the right. The majority of colonies are edited on all alleles, with only some being heterozygous editing. And then when we compare this with the essential line, which we can see on the right, The data shows that the majority of clones have absolutely no editing, with only very few colonies having either heterozygous or homozygous editing. So these two here. So once we have the restriction digest assay result, we can analyse the number of nucleotides inserted or deleted in a subset of those colonies. We once again perform a PCR, however this time one of our primers is fluorescently tagged. The resulting products are analysed by capillary electrophoresis and the size of products is determined. Comparing the colony data with the parental control, which here you can see in the top, is 417 nucleotides, we can determine the number of nucleotides inserted, deleted at the site of editing. So here's the data for a target with a copy number of 2. And as I said, comparison of the fragment size with that of the parental enables us to determine the size of the indel on each of the alleles in these last 2 columns. So here are the results collated, and in the independent line here at the top, the colonies that returned as having homozygous editing events by the restriction digest assay were actually confirmed as this by fragment analysis. And the colonies that were classed as heterozygous by restriction digest, when we looked at them by fragment analysis, we found that they were edited on all alleles. This means they may possess indels that have retained or reconstituted the restriction digest site. In the dependent line, here at the bottom, the colonies that returned as having heterozygous editing were confirmed as heterozygous by fragment analysis. They retained a wild-type length fragment. However, what was interesting is the colonies that were homozygous edited by restriction digest, and in this example there were actually only 2 colonies, they retained a wild-type length fragment and therefore should retain gene function. So we can also sequence the PCR products by Sanger sequencing. To do this method, the best data is obtained from cell lines with lower gene copy numbers. For higher copy number genes, we could obtain the data by topocloning of the alleles, where we can sequence each allele independently. In this example, the restriction digest assay had classified the colonies as having homozygous disruptions, but the fragment analysis data returned a fragment of wild-type length in all cases. Sequencing of the edited region revealed that substitutions had occurred within the edited region that had disrupted the restriction enzyme recognition site without changing the overall fragment length. Therefore, they truly were homozygous disrupted, but were likely to have retained function. So these examples here, in the DNA sequence, you can see that we've underlined the restriction digest site, and in these alleles indicated in red, you can see where the substitutions have occurred. The other issue we have is that fragment analysis does not produce a round number, and manual rounding of figures is required. Sequencing of a few samples enables us to check the fidelity of the rounding. As we can see in clones A and C, which is this one here and this one here, the fragment size was rounded down, but after sequencing, we see that it should have been rounded up. So the fragment analysis assay picked up a 15 base pair, and when we did the sequencing, we actually saw a 16 base pair deletion. However, with clone B, we can see that the fragment analysis data picked up a 47 base pair deletion, which we could also see by Sanger sequencing. So using sequencing data from a few colonies ensures that the assumptions made in the fragment analysis assay are correct, and any required adjustment can be made to all samples. For this data set, we realised that any small deletions need to be rounded up, whereas large deletions need to be rounded down. So we've also used this assay to follow up on data which we obtained in one of our target validation projects. In our alliance with H3 Biomedicine, we were following up observations from the Tuvson and Barbacid labs that showed that CRAF is required for the initiation and development of mutant KRAS-driven lung cancer. This project involves studying CRAF dependence in 21 non-small cell lung cancer cell lines consisting of 13 mutant and 8 wild-type lines. Stable doxycycline-inducible shRNA expressing lines were generated and the effective depletion of CRAF was studied, And here I'll show you an example of one of the wild type lines. You can see in this western blot on the left that CRAF knockdown was good for all of the shRNA used. However, knockdown of CRAF had little effect on proliferation. In this proliferation assay, which is expressed as induced cells relative to uninduced cells, we can see that none of the shRNA to CRAF, which are these ones shown as the orange bars, have any effect on proliferation. And this was replicated across all of the wild-type KRAS lines. However, the results obtained for the KRAS mutant lines were more ambiguous. Again, we saw good knockdown of CRAF with the shRNA, equivalent to what we saw in the wild-type lines. And overall, the KRAS mutant cells appeared sensitive to KRAS depletion, to CRAF depletion. However, there were situations, such as this one, where we used A549 cells, which have a KRAS mutation, where an shRNA, in particular in this one, number 784, which is this one here in the middle, successfully depleted CRAF but had no anti-proliferative effect. So as I said, here on the left we can see the knockdown is equally good with all three shRNAs, but when only shRNA number 1 and number 911 had an anti-proliferative effect. And we were unsure whether these were true effects or whether they were off target. So we attempted cDNA rescue experiments with these cells, but these were also inconclusive. And soon after we abandoned this project, the CRISPR-Cas9 system was discovered. And given the promise of CRISPR to overcome threshold events, which we thought may be a contributing factor to the ambiguous data that we obtained in the KRAS mutant lines, we attempted to validate CRAF using single sgRNAs in an array-based assay similar to that used for shRNA. So using, again, A549 cells, we infected the cells with the P-lenti CRISPR vector containing an sgRNA against CRAF and then monitored proliferation using an Estin incucyte zoom. So the data is shown here in this graph on the left. The parental line is this one here in dark blue. We have a negative guide RNA which is targeted to the ROSA 26 locus in light blue. And we have 5 guides directed to CRAF, which are these lines here in orange. As you can see, we did see an anti-proliferative phenotype with the CRAF guides, but this was incomplete. And growth rates of the targeted cells appeared to be returning to normality during later phases of the study. And this was the same as I showed in the model data earlier. So we predicted that these results were due to the occurrence of in-frame editing in the CRAF gene. So, using the Pathfinder Target Essentiality Assay, we were able to recover approximately 30% of colonies that had frame shift deletions in CRAF, which confirmed that the data that we got with the shRNA 784 in earlier experiments was the true result. KRAS mutant cells can survive and proliferate without CRAF. So here we have a simple flowchart documenting the workflow that's involved in the assay. We start with selection of the cell lines and the guide RNAs if it's required. And those chosen cell lines are infected with P-lenti CRISPR containing the sgRNA, and then they're cultured for 2 to 3 weeks and plated for single cell colonies. We then perform the restriction digest assay. For a copy number of 2 to 3, we do this typically on about 384 samples per cell line per target. And then once we have those results, of these colonies, approximately, say, 48 per cell line per target are analysed by the Fragment analysis assay. Now these dark blue boxes outline the standard package. However, there are a number of ways that we can modify it to suit your needs. At each stage, we can scale up the number of colonies plated or the number of colonies analysed. And that's what we would advise if your gene has a high copy number. However, we can also perform additional analysis such as Sanger or topo sequencing, and it's also possible to keep some of the colonies for further downstream analysis on other things such as proliferation. So for a cell line that single cell dilutes well and grows at an average rate, we'd expect the standard workflow to actually take about 4 months. And also, here's the list of cell lines that we've pre-evaluated for use in this assay. However, it's possible to evaluate cell lines not on this list for suitability. So, what else is Horizon doing with CRISPR-Cas9? A powerful application for CRISPR-Cas9 is in functional genomic screening. Here we can use a pooled approach similar to that used for shRNA screening. We've got a number of in-house libraries against targets of high interest in oncology, as well as whole genome libraries, and we also have the capability to produce custom libraries for our clients. These libraries are deployed with Cas9 in resistance, sensitivity, or even synthetic lethal screening, where we can use next-generation sequencing for sample deconvolution and target identification. One further application for Cas9 that we're exploring here at Horizon is the repurposing of the Cas9 enzyme for use in transcriptional control. In this system, the enzyme has first been inactivated at both of its nuclease sites so that it simply has a sequence guide and binding function. Already out there are several generations of this technology which can be used for either transcriptional repression, or known as CRISPR-I, or transcriptional activation, which is known as CRISPR-A. We're using these tools in both target validation and target ID large-scale screens, and they offer new opportunities in functional genomics. Horizon's Pathfinder research services includes precision gene editing, isogenic cell line assays, cutting-edge screening platforms, and in vivo models to help solve your research and drug development challenges. We work closely with you to design the studies that best answer your scientific questions. We offer integrated solutions to support drug development programs, including custom in vitro and in vivo model generation, target identification and validation, both in vitro and in vivo efficacy studies, identification of responsive or resistant patient populations, mechanism of action and resistance mechanism studies, and drug combination screening. And of course, we also have all the typical offerings expected to support oncology drug discovery programs, such as pathway analysis via Western's or ELISA, and we also have the capability to do live cell imaging. What's most important is that we're not a typical off-the-shelf CRO. We actively input into a client's project design and data analysis, and our services can be accessed either individually or as part of an integrated project with our other sites worldwide. This enables us to support projects of any size and aim. We apply strong scientific principles to all of our projects and try to find solutions that fit your needs. We put the emphasis on using the right model systems and study design to test your hypothesis, not just project execution.